So, my benthic career began during my undergraduate days at Cornell. Don Hall and Bill Cooper put together this really fascinating experimental ecology study. Don was a professor at Cornell, Bill was a professor at Michigan State. They invited me to help come out there and pull seines through some of these ponds. It was a classic experiment of nutrients versus predation. It was the longest paper, as far as I know, ever published in L&O. It became a citation classic. But for me, the Cornell pond study was an introduction to the benthos. Here's where I got my interest, and particularly dragonflies. I could recognize early on that dragonflies represented many, many possibilities for research back in the mid to late 1960s. And it's kind of interesting. On this project, there were 3 students involved. Well, there's more students than that, but 3 of them ended up doing their dissertation work on dragonflies. I was one of them, and another one is here. Where are you, Dan? Dan Johnson, back there, returns after 8 years. Dan's retired now, but Dan is the one who kept dragonflies throughout his whole career. I fell by the wayside a little bit, worked on a few other things. So, what's not to love about dragonflies? What fascinating animals. You just saw one of them in action. They're spectacular prehensile labium. They can flick out like the tongue of a frog and capture their prey. They're jet propulsion, really neat animals. And their adults are kind of neat too. They're also incredibly voracious predators, yet they're beautiful. And we love to collect them. But that was my experience as an undergraduate. For graduate school, I went off to the University of Georgia. And I knew I wanted to work on dragonflies. All I needed was a pond with lots of dragonflies. And I found such a pond at the Savannah River Ecology Laboratory area in the Savannah River plant. I also found a fellow by the name of Dave Coleman, a young assistant professor who would be my major professor. And he eventually became a highly acclaimed soil scientist, but he was just a young guy, not too much older than I was at the time. I like to think of him as a terrestrial benthologist. But I really wanted to be an experimentalist, just like Hall and Cooper. This was the modern way to go back in the 1960s even. So you guys doing field experiments today, you know, this goes back a long ways. I was pretty fortunate to be able to get a few good papers out of my dragonfly work at University of Georgia. These are my best papers. Whoops, didn't mean to do that. The one at the top here is on comparative dragonfly life histories and dynamics. Then there's one on dragonfly production, and then there is one on the experiment that I did. And just briefly, on a very low budget, I managed to construct these pens along the shoreline of a pond. I managed to fool certain kinds of dragonflies to not lay their eggs in certain pens. And then I wanted to look at the response of other dragonflies that I allowed them to lay their eggs. The results turned out pretty well. I'm not going to dwell on them. But, you know, it worked. But I also did another study, this comparative dynamics and life history study, in which I looked at the larval life histories of the dragonflies. And it turned out they were very synchronous life histories. And there was temporal separation between the species. And not many people were showing this kind of thing, even back in those days. There were a few people working on streams that were doing it. Rosemary Mackay, Alan Hildrew. I don't know if Alan's here this year. But this was pretty neat. It suggested a reduction in a resource partitioning through time. The thing about dragonflies was, in these ponds, they were really high densities. It's hard to imagine predators with densities of more than 100 per square meter, but there were. And because the densities were so high, you could sample them with an Ekman grab. You didn't have to use a big box sampler. And furthermore, they had constant survivorship curves. And not many people have been trying to describe survivorship curves for aquatic insects And John Lawton is the only one that I can think of from England that was doing this. In fact, it was with damselflies. But probably the paper I got out of my dissertation that had the biggest influence in my career was one on secondary production. If animals develop synchronously, they show a synchronous growth pattern and a survivorship rate that's constant. It really doesn't even have to be constant to do this. One can calculate production. And here's where I started learning from Tom Waters, his publications from the late 1960s and early 1970s. And I just thought doing this from field data to be able to estimate a flow of energy or mass through time was really neat. You didn't have to even do an experiment. And so we ended up with really high production values for just 3 of the dragonfly species. There were several others. Probably the total production of these predators, and these are strictly predators, they don't eat anything else, was probably close to 10 grams per meter squared per year. That's really high. Well, the only problem was that their prey species, you see down in the bottom right, and I know you can't see that axis, but this is less than 1 gram per meter squared for the prey biomass. How can dragonflies produce so much? So we had an example then of the Allen paradox, which has been shown by Allen since 1951, and there are other examples as well. But what it meant was that the prey biomass had to be turning over more rapidly than anybody thought was possible at the time. The generality was the P to B ratio for aquatic invertebrates at the time was more like 10, maybe 15. It just was kind of unheard of to have such a high turnover. So, the biggest lessons I learned from my dissertation were, I think, were that we can understand a great deal about how populations and communities function in nature without experimental studies. Experimental studies are great, but you can't do them everywhere. And simply by going out into nature, by using quantitative sampling methods and quantitative analysis tools, you can measure an awful lot of things that are very meaningful and help you understand aquatic ecosystems in ways that you really couldn't discover from experimental studies. But also, I became very intrigued by methods of estimating secondary production, and this defined much of the rest of my career. What I whizzed by there, the last thing was I became very intrigued by the size-frequency method, which was originally called the Hines method because it was developed by Hines and Coleman back in 1968. The only trouble was Hines didn't get it quite right, as he probably pointed out in the very first Award of Excellence talk. And he talked, I remember to this day, this was back in 1988, about how you can go through science and don't be worried about making mistakes. And Andrew Hamilton was there within a year to correct his method, and he wrote a beautifully written paper in 1969 on corrections to the method. But Andrew Hamilton got one small thing wrong, and that was to adjust the method for multivoltine species. He suggested you multiply by the number of generations per year. And I was willing to accept that, and I was trying to understand the method, and then I realized sometime late in the 1970s that this wasn't really quite right. What you had to do was to adjust the calculation, the basics, what we then called the size frequency calculation, by not looking at the number of generations per year, but by looking at the development time in the larval stage. And so this may seem like a fairly subtle difference to you as benthologist or aquatic scientist, but it was important from at least one point of view, and that was that it meant that P to B ratios could be much higher than we'd previously envisioned. They could easily be greater than 10, 15, they could even be greater than 100. And so that implication, I think, helped at least change my view on how these systems work, and it really has significance for nutrient cycling and other aspects of turnover of biomass of small animals. And in a sense, it became a kind of an explanation, at least a partial explanation, for the Allen paradox. So all of this happened after I'd moved on to my first job at Georgia Tech. I moved there in 1971. That last paper wasn't published till 79. But in the middle of that time, I got involved in coastal plain rivers. The Satilla River, a Blackwater River in southeastern Georgia, you can see on the slide. And the reason I got interested in it, and this was my first adventure into flowing water, but the reason I got interested in it was I had heard there was wood in these rivers, and they were colonized by lots of aquatic insects, not something that the benthologist usually thinks of. And sure enough, I went down there and investigated, and they were just covered with all kinds of things. And I said, you know, we got to study this. This is really pretty neat. And so I got a small grant and a fairly minimal budget, and we started doing some studies. We published 3 papers, my colleagues and students and I. One was on distribution of production among habitats. Another was on well, it was an overview, but it focused on what fish ate. And a third one, which was in JNABS, on drift dynamics and where the drift came from. But there were tons of things on these snags. This is just a partial picture. I don't even have the mayflies, the stoneflies, and the beetles on this collage. But you can get the idea. The big predator was down there on the right, the Helgramite corydalus. And the most important paper was the first one, the Ecological monograph. It was 39 pages long. I said after I was done, never again. It was such a long paper, and I never wanted to write one that long again. But there were a lot of tables and figures in there, but we can kind of summarize the whole paper in this diagram to the right. Basically, what you see there is the highest production. It shows the highest production being on the snags. Also, the highest diversity of insects being on the snags compared to the sandy benthic habitat of the main channel and the muddy habitat of the backwaters. So this was the real hotspot of productivity and diversity in this system. Now, at this point, before these papers were, well, I'm sorry, I'm getting ahead of myself. Let's go on to the fish. It turned out that several species of fish, we were able to sample fish every 2 months from the river. We did gut analyses, and for many of the species, it turned out they obtained their food from invertebrates that lived on the snag habitat. The drift data showed exactly the same thing. 3/4 or more of the drift biomass was from the snag habitat. So these 3 independent sources of information show that submerged wood was really important to the ecosystem functioning of these coastal plain rivers. And this encouraged me, along with Judy Meyer and Bruce Wallace, to begin another study on coastal plain river, which I'll get to in a minute, but I just say that as a transition here, and point out that I got involved with another project with Bruce Wallace, actually my first project with Bruce Wallace, who was on my committee actually at the University of Georgia. And he was over at Georgia Tech in the late 1970s, and he gave, you know, a typical Bruce Wallace talk, you know, caddisfly life histories, what they eat. He was very good at dissecting apart their little guts and seeing what they ate. So we went out to a French restaurant that evening, and I said, Bruce, you ever think of doing production with this kind of data? Or is this quantitative samples? He said, yeah, it's quantitative samples. I said, why don't you do a production study? Just take this data and do production. He says, well, he was a little hesitant. He says, well, maybe if you help me. I said, okay, send me your data. I'll do all the calculations for you. So he did, and he sent me the data on the feeding as well. And so we were in the position to ask the question then, what food types were responsible for production of net spinning caddisflies in mountain streams?